That's it. Can I? Yes. Well, it's a great pleasure to me to be here to talk to you. And then I will present my work called the glutathione transferase GST polymorphisms, influence cisplatin elimination and toxicity to treatment in patients with squamous cell carcinoma of head and neck. I'm Eder Pincinato. This work is results from my doctoral degree developed in the University of Campinas, and I'm also a professor at Mackenzie Presbyterian University in Brazil. Well, that's I'm planning to present to you. I will start my presentation doing some introduction about head and neck cancer that responds to 560,000 new patients worldwide annually, and is responsible about 200,000 deaths annually. That's a huge number of patients that die with cancer, head and neck cancer. The mainly location of this type of cancer is oral cavity and pharynx and larynx, responding at about 90% of these patients overexpress EGFR, showing that you have some genetic important information in these patients. About 40% of these patients have overall survival about 5 years, and you have here the main risk factors to develop the head and neck cancer is high consumer of alcohol and high consumer of tobacco. Also, HPV is an important risk factor to develop the head and neck cancer. Mainly, the main people who do have this type of cancer is men. Men, it's 2 times more available to develop this cancer. The treatment to this type of cancer is surgical resection when the patient comes to the hospital with low levels, low stage, and this surgical resection is considered the only curative treatment for this type of disease. But when the patient presents high stage of the cancer, we also treat this patient with radiotherapy associated with chemotherapy. And so in chemotherapy, we also applicate cisplatin as a chemotherapy agent. Cisplatin is considered an alkaline antitumoral cytotoxic agent. And post-infusion, about 90% of cisplatin binds to protein in blood and have rapid diffusion to a lot of tissues. For example, to kidney, to lung, to uterus, and the liver. After this rapid infusion, cisplatin enters in the cell with utilizing copper transporter. And then we can get the renal extraction of cisplatin, and about 80% of cisplatin being eliminated in urine 24 hours post-infusion. Once inside the cell, cisplatin can make a duct with DNA. Then with this duct, the cell can activate your mechanism for apoptosis based on caspases. Cisplatin also can be conjugated with glutathione mediated by the enzyme glutathione transferase that forming high hydrophobic agent that can be more easily eliminated within our organism. Cisplatin also can be associated with ATP7A and ATP7B and then put through the cells, out of the cell. The main problem to use cisplatin that's high effectivity to this type of cancer that cisplatin can induce adverse effects to the patient. main causing nausea and vomiting, myelosuppression, ototoxicity, and nephrotoxicity. And we also see in the treatment of these patients that when we treat people with the same clinical condition and the same laboratory condition, some of these patients respond differently using the same dose. To explain this kind of response, we can respond relation to the genetic polymorphism. Polymorphism can respond this difference of these patients. Then we have some polymorphism being demonstrated to the receptor of the cell surface. We have also described some polymorphism related to repair mechanism of the cell, and the main focus of my work is to study how these polymorphisms in glutathione-transferase detoxification can influence in this type of resistance of treatment. Then a lot of questions can be done related to this polymorphism. Is there difference in therapeutic response associated with the glutathione-transferase polymorphism? Also, is there influence of these polymorphisms in cisplatin adverse effects related to the treatment? Or also, is there a relation between cisplatin pharmacokinetic and this polymorphism? In scientific communication, we have a lot of papers, but they are also controversial, or there are few studies related to the elimination of cisplatin related to this polymorphism. Then, the aim of my work is to verify if GST-M1, GST-T1, and GST-P1 polymorphism in patients with head and neck cancer are associated with responsive rate to treatment with CDDP or cisplatin, associated with radiotherapy, associated with toxicity to the treatment, and associated with CDDP pharmacokinetic modification. To reach this goal, we took 145 outpatients with squamous cell carcinoma of head and neck that do treatment in oncology clinic at Unicamp Hospital. It's a tertiary hospital. Then we included 145 patients, but we have excluded 55 patients. We lose these patients by changing in therapy or with the death of the patients, or changing in some conditions that they are unable to use cisplatin as main treatment. These 90 patients that we are including in our study receive 35 sections of radiotherapy and 3 cycles of infusion of cisplatin, varying from 75 to 100 milligrams per square meter on day 1, day 22, and day 43. In this cycle, in each day of we received infusion, we took urine in 3 periods. One period varying from 0 to 12 hours, then 12 to 24 hours, and 24 to 48 hours. The urine were then extracted and analyzed by HPLC. Here I put some, that's difficult to see, but I put some example of chromatography of these patients. We can, by HPLC, we can see the first peak of cisplatin, and the second one, the internal standard used to analyze this condition. Also, we took blood sample for these patients. We extracted DNA, and with the DNA, we have done GST-T1 and GST-M1 analyzed by multiplex PCR, and GST-P1 was analyzed by PCR, followed by enzymatic digestion. The fragments were done in agarose gel, and them revealed with ethidiobrometum. Well, our patients were here characterized. The mean of age of our patients were about 55 years, 0.6 years. Main group are formed by males. About 92% of the patients were males, were Caucasian, and were heavy smokers and very heavy drinkers. Very heavy. And this is a strong association observed in our group. The main tumor local was in pharynx and followed by larynx. Here we have some different from literature, that the main observed in literature is oral cavity. But when the patients enter in our group of study, they are with a high stage of tumor. Then these patients with oral cavity has treated before come to us. And they present, many of our patients present high stage of the disease. And the Karnofsky's performance status was about 860 in mean. At the beginning, yes, at the beginning. Some of them we need to take out about the KPS. Here here I presented my distribution of these patients related to this polymorphism. Main patients of our groups present null gene for GST-M1, and main patients with our group present GST-T1 presence of the gene. And we have also same distribution for wild polymorphism from GST-P1 and heterozygote polymorphism from GST-P1. This distribution is also observed in other population. That shows that our patients are representative for that we observe in the world. When we analyze the genetic polymorphism associated with vomiting, we can see that patients with null genotype for GST-T1 present less high grade of vomiting compared to patients with the presence of the gene. In the first observation, this is some strange results that we not expected. And when we compare GST-P1 gene, we found that patients with wild genotype has less high grades of vomiting compared to patients that present allele variants. Thus, we can observe that null genotype to GST-T1 protect patients, and variant allele to GST-P1 induces high grades of vomiting in our patients. When we studied the ototoxicity of these patients, we also see that patients that have null genotype to null polymorphism to GST-T1 present less have any grade of ototoxicity compared with patients that have the gene. The same observation that we had last. And also, when we studied GST-P1 105 vol, we observed that wild patients with to GST-P1 have less high ototoxicity compared to patients with variant allele. Again, null genotype may protect patients related to GST-T1 polymorphism, and variant allele induces high grade of ototoxicity related to GSTP1 polymorphism. When we evaluate the nephrotoxicity to these patients, we observe the same profile. The patients we present null gene from GST1 have had less difference after and before treatment related to clearance of creatinine, and observed also with chromo 51 EDTA. And also, once again, we observe that null genotypes protect patients in relation to nephrotoxicity related to GST1, and variant genotype have demonstrate higher nephrotoxicity when compared to wild genotype related to GSTP1 polymorphism. It's so clear, sorry. But when we analyze cisplatin elimination kinetic, we also observe that the main concentration of cisplatin is eliminated in the period of 0 to 12 hours, associated in the period to 24 hours, responding to elimination of approximately 94% in the first 24 hours. Again, this result is compared to the literature. Then, when analyzed total cisplatin elimination related to the genes, we observe that, again, patients that don't have the gene, patients that have the null genotype for GST1 have higher elimination of concentration of eliminated cisplatin. When we analyze this together, we can summarize the patient with toxicity related to toxicity that we observed that GSTP1 allele variant increased the risk to toxicity, increase in vomiting, increase in ototoxicity, and increase in nephrotoxicity to these patients. Then GST1 null genotype protects the patient that will observe less vomiting, less ototoxicity, and less nephrotoxicity when compared to patients that have the gene. Associated with these results, we also see that GST1 null genotype eliminates more total CDDP, contributing to less toxicity observed in these patients. To hypothesize the results, that's some kind of strange to us. We hypothesize this condition. When patients present GST1 null genotype, more glutathione. Sorry about this slide is in Portuguese. I don't have too much time to translate, sorry. But when we have patients with GST1 null genotype, more glutathione is available to patients with wild genotype to conjugate more cisplatin. Then, when they conjugate more cisplatin is put out of the cell, diminishing the toxicity observation. Or we can see less vomiting, less ototoxicity, less nephrotoxicity. But we can't find that this interfere in therapeutic effect. We didn't see no alteration, no difference between the groups relation to therapeutic effect. Sorry?
These genes are mainly in the kidney, in the normal cells, or are in malignant cells?
Malignant cells. this kind of gene, it's distributed in all of the cells. And it's the mechanism of detoxification of canabiotics. And sorry.
Based on your theory, because if it's in the kidney cells at the nephron, when you have the GST1 overactive, and now you increase conjugation, increase conjugation of the cytoplasm, that's why it's out of the cell.
Yes.
Eliminated. So that's why you increase elimination, and the cell is not toxificated.
Yes.
It makes sense to me.
Mm-hmm.
And I think the same thing will happen because when you increase elimination, you decrease the level
Of toxicity.
so you decrease the toxicity. That's why you see vomiting is less,
Nephrotoxicity and toxicity. Yes.
But when we talk about the malignant cells,
Mm-hmm.
it doesn't fit for me because if you still increase conjugation and take it out of the cell, this means that
less therapeutic effect it's observed.
Let's take the comments afterwards. Yeah.
Mm-hmm. Yes, we asked for ourselves that why this can be observed. We observed that there is no difference between these groups related to therapeutic effect. Then I'm not able to respond now to you why this mechanism can be done. We have done this type of question because for me it's also the same idea that if I put the cisplatin out of the cell, less effect I will done, less duct with DNA are formed, and less effect I can see. But again, we can't observe this difference between the groups. It's a small group. We start with 90 patients. Maybe if I can put the pathway, I can analyze this better. But that's the number that we can work with it. Mm-hmm. And there is on paper that show the same results, not with head and neck cancer. If I can't remember now, but this type of results was also observed from, I think, testiculous cancer. Yes, I think yes. And our theory said that if these patients, we have these results, but we have low group of patients. But we also have another results that show if we associate the three polymorphisms that we observed that patients that have null genotype from GST1, null genotype to GST-M1, and wild genotype for GSTP1, these patients has the lowest toxic effect from the drug, with no difference in therapeutic effect. But this group is only formed for 2 patients, and then I can do this statistically to this group. It's too low. And then we hypothesize that this can be done with these patients, that if they don't have the gene, more glutathione can be available to the wild gene from GSTP1 to detoxificate more cisplatin and diminish the results of toxicity. Well, then we can conclude that cisplatin is approximately 85% eliminated in urine in a period of 0 to 12 period. CDDP detection by HPLC is a simple and powerful tool that can be performed to evaluate the pharmacokinetic of the drugs. And GSTT1 null genotype and GSTP1 wild genotype protect the patients in relation to the toxicity induced by CDDP, or cisplatin. And patients with head and neck cancer, squamous cells, with inherited distinct abilities for cisplatin metabolism associated with GSTT1 and GSTP1 polymorphisms exhibit distinct toxicity to treatment and urinary cisplatin excretion, and may receive more attention during treatment with the purpose of avoiding the side effects of the chemical agent. It's the beginning of our study, and in the future, maybe I can be able to look at the polymorphism of the gene of the patients and then adjust the dose based on the genetic of the patients. That's it. I would like to thank you for your attention and for your patience.
Do you have some questions? Yeah.
Hello. Yeah. Question about dosage. You said there was 35?
75 to 100 milligrams per square meter.
35 sessions. 35 sessions.
Yes, 35
and
sections.
Why 35? And was they given therapy each day?
Yes. The patients becomes the first day they receive cisplatin infusion, and then subsequently they receive each day 2 grams of radiotherapy per session. And
so
then
this is standard therapy, standard dosage for these patients.
Sorry?
So this is standard dosage for these
Yes,
patients? Yeah.
yes. The patients only don't receive radiotherapy on Sundays. And then, on Saturday and Sunday. Then on Monday they receive again the radiotherapy.
More question? Ayman?
Thank you. This is excellent study, by the way. This is really high quality study, and you are right, because that study basically aimed to, it's like a phase 1 study. It's basically just to identify the toxicity and adverse effects of the drug, and then relate that to the genetic structure. So I'm not worried about the clinical outcome here, because I don't think this was designed to see it. It's just simply because the outcome or the activity, there is lots of factors. The level, the degree of the malignancy, all of the things are there. So I'm not worried about that. But I think the beauty about this study is just explains exactly, beautifully, how the nephrotoxicity and the toxic effect of cisplatin can be different from one to one by just having this type of gene. The gene here, that CSTT1.
Yes,
GSTT1. I think you elegantly explained that. It's beautiful, because it shows that if I have this gene, this means that the drug comes to my nephrons, or the lining of my kidney, where the nephrons, so where the toxicity, the drug come here, but it's less conjugated, so it gets out of the cell.
Yes.
So there is no accumulation of the drug, there is no toxicity. It explains everything. It explains nephrotoxicity is lower than that, and also it explains that you have more elimination, because cisplatin is basically eliminated renally, so you have more elimination, less levels, so now you don't have toxicity, vomiting, there is no nephrotoxicity or even ototoxicity, because again, the same things like aminoglycoside.
Yes.
It goes to the blood and goes to the inner ear and then begin to cause all of things. It makes lots of sense, frankly speaking, when I see something like that. But then what you get out of that? Do you get out of that you need to go do some screening for patients a priori before you start treatment, to identify patients who might be even more expected to show toxicity or less toxicity, and try to find some other way of maneuvers to decrease toxicity if I have a patient who doesn't have this protective gene and have that other gene. So what's your plan out of that, other than just saying that these are the genes that are associated? So if there is something that you can do further to make it more safer for our patient to be treated.
Yes. Patricia will present another results in this related to this group of patients that she uses another preventing mechanisms that are not looking for the genes. She also presented her results. But I think that I can look for the genes of the patients, and then we can separate the groups. Then I think the next step is to analyze these groups, and for example, I will separate these groups according to the gene, and then I will make some adjustment of the treatment, and then I can see if my adjustment is preventing the nephrotoxicity and all toxicity, and not interfering in the overall treatment of the patients.
Maybe I'll just make some suggestions, if you allow me for 5 seconds. I think also maybe very interesting in the future, if you use patients who are Caucasian, you mentioned that. So I see that all of them are one category. Maybe, I'm not quite sure what the expression of these GSTs, genes in different ethnic backgrounds. So you talk about Caucasians in Brazil, but maybe you have some other ethnic background. I think there is lots of, maybe African American. Maybe it might be interesting to see if there is difference in the gene expression between different, population. This is one thing. The other thing is that you did some kinetic study, which I don't like it. I have to say that you didn't say kinetics. You just said you measured the drug. It is there, but I really wished if you have in the future, if you had different samples, if you can do kinetic study, you can follow the concentration and elimination in the blood.
Yes.
Not even the urine. Urine will show you what happened in the kidney. Yes. But just to see exactly, is there something like that? Then when you have another factor, it might be able to explain that I'm going to increase the elimination. I'm going to do something that will decrease the toxicity this way.
We have some difficulty to do this kinetic study, the perfected kinetic study, studying the blood of the patients. But they are outpatients, and then I can't take blood sample of these patients to analyze cisplatin in the blood. That's because we have done it in urine. That is not perfect, but
urine is okay also. You can do some kinetic study.
Yes. But the next step again in our group is to take these patients to put in the hospital, and then I can take blood from hour to hour, or about 2 hours, 4 hours, 8 hours, and then we can do this curve, the total elimination.
I think we soon run out of time here because...
so only one question, Prof.
Okay.
This time duration, sampling time, how you determine that?
Excuse me, I didn't...
Sampling time, the collection of blood sample. What is only
the
one sample or interval?
To the urine collection?
Yes. Urine and blood, both.
I took blood sample only in the first visit of the patient in the hospital to enter in our group. And then with this sample, we extract the DNA and make the polymorphism genes.
No. My question is, you are admitted cisplatin in the bolus dose or infusion dose? If you are admitted in the bolus dose, what is the sample intervals? If you are admitted in the infusion dose, what is the sample intervals? How you collected the samples? At what time you collected the samples?
The samples, the blood samples was collected after the infusion. The infusion was
the infusion, right?
Yes.
How duration of infusion?
Yes, they enter in the hospital. They receive 3 liters of saline in infusion.
3 liters of saline.
Yes. Then they receive infusion of cisplatin during about 1 hour and a half. They receive
it's possible cisplatin. 3 hours you can instill 1.5 hours durations?
Yes. And then they receive 1 liter in saline, infusion of saline. Then the patients go out, and I give to collect the urine. I give the patients some bottle to collect urine for 24 hours.
if you are collecting the samples,
I think I have to stop you. I'm sorry to interrupt there, but there's a lot of others who have to come in.
it's okay. Basically, I worked in the.
You can talk after.
No issues. If okay, I don't want comment.
I think we shall
Thank
do
you.
that. But anyway, it was a perfect presentation and a perfect paper, actually. So it was no doubt for you. I don't understand why you are afraid. No reason for you to be afraid of that. A very good presentation and a very nice paper. And you know, actually, we should have a certificate, but